think we're good. Okay, good. Okay.
Yeah, we're gonna start.
We're starting. All right, everybody. So I just wanted to start with taking you behind the scenes to the collections, if you haven't been there before. So if you were to walk into the Ornithology Department and you were to go to what we call our main range, which is where most of our specimens are kept, you would see a room like this. You would go into one of these open rows and you would see a lot of cabinets stacked on top of each other. And then if I were to open them and pull out drawers, you would see a lot of bird specimens. So we've got about 100,000 of these bird study skins of birds from all around the world. This is mostly Crassiformes, hornbills. So they're taxonomically arranged approximately. And then as a scientist, I draw my inspiration from this diversity that we can see in the collection here. So what really got me excited is learning why did this bird diversity come to be and how is it changing as the world is changing. Just a few more stats about the collection. We've got, as I said, about 100,000 of those study skins. We also have about 17,000 complete bird skeletons, which is one of the largest bird skeleton collections. We have a very strong paleontology department, also the liberated tar pits. People think of that as having a lot of saber-toothed cats and mammoths, but there are actually more bird bones that come out of the tar pits than anything else. We've got like millions of bird bones, many of which are unidentified because it's very difficult. But having a reference collection is really important for that. We've got about 3,000 fluids preserved specimens. So think birds pickled in jars. And these are increasingly being used for science because it used to be, this is how you have access to the inside of the bird. The study skins are just stuffed with cotton. If I was wanted to study, the digestive system or the muscles, I would have to dissect out one of these. These days, we just pop them in a CT scanner, and you get the entire 3D reconstruction.
Wow, that's great.
Which is very cool.
That's very good.
And ever since about the 80s, whenever we prepare a specimen, we also will take little samples of the tissues, usually heart, liver, and muscle, and we preserve those in a cryogenic freezer to make genetic analyses easier. We actually can do genetics with the study skins in any of these specimens. It's just much easier with good high quality tissue samples, and we can do more with that. So that gives you, so we have over 123,000 specimens in total in the department now. Where are our specimens from? Well, California, of course, has the greatest number of specimens. You can imagine, especially around Los Angeles. But we also have large collections from East Africa, from Brazil, from Argentina, various places in the Pacific Ocean. So we really have a worldwide collection. And why do we have specimens? Well, that's where the curators back in the day, mostly actually in the 60s and 70s, went on these big expeditions. So that's why we have specimens from there. And so as I mentioned, as a researcher, I really want to understand why and how did the diversity of birds come to be. And of course, that's a very large topic, so I have to break that down into a few different categories to focus on. The first category that I focus on is plumage coloration, so how and why are birds the colors that they are, and that's most of what I'm going to focus on in this talk tonight. But I also study host pathogen coevolution, meaning how are birds evolving in response to pathogens, and then how are the pathogens evolving to be better at infecting birds and vice versa. And then finally, urban evolution. So how are birds evolving due to kind of human-induced changes in the environment, especially the urban environment? So the main group that I study for the evolution of color is this group here, which is the tanagers. So this is a rainbow of tanagers, which illustrates why they're so useful for studying the evolution of color. You have virtually every color represented by the single family of birds, so we can test a lot of kind of evolutionary and ecological hypotheses using them. But there are also many species that are more drab colors. So they might be blacks or browns, and really having both colorful and less colorful birds make it so we can understand when are birds colorful, when are birds not colorful. A lot of this work really started during my master's at San Diego State with Dr. Kevin Burns. And the first thing we do was actually to construct a phylogenetic tree, meaning a history of the evolutionary relationships amongst tanagers using genetics. And there are currently 382 species of tanagers. This number is kind of gone up consistently since I remember when I was a master's student, it was 365. And I was like, that's perfect. We can make a calendar, tanager a day, it'd be great. Now we have 382, that's okay. They make up about 10% of songbirds, which is a huge percentage of the total bird diversity. And just looking at this kind of arrangement, you can see not only are they very colorful, but they have a huge diversity of bill shapes. They live in virtually every environment in Central and South America. So they're really useful for understanding bird evolution generally. Fun fact, Western tanager you might be familiar with around here. Not a tanager, actually a cardinal, genetics told us. So we actually don't have any tanagers here in Los Angeles, and really not really in the US. Some of the seed eaters like get just in the very south part of Texas, but for the most part, tanagers are just in Central and South America and a little bit of Mexico. So understanding why are birds the colors that they are, there are two hypotheses that have generally been proposed. One is that birds evolved due to natural selection. So this is selection, for example, to be cryptic in their environment so that predators can't see them. Alternatively, you might expect that colorful birds have evolved due to sexual selection. So that is selection to either to attract mates or to fight off rivals. And so this, this whole debate goes all the way back to Darwin and Wallace, who, Darwin proposed natural selection, Wallace proposed sexual selection. And so really this question of which one is more important in driving the evolution of bird coloration has been around for quite a long time. And how can we understand that? Well, we can actually make use of male and females and the fact that in some species of birds, males and females are the same color. In other species of birds, males and females are in different colors. And so we know that these species share the same evolutionary history. They share the same genetics. They share the same environment, but there might be some behavioral differences. And so by understanding what those differences are, we can understand, okay, is it sexual selection or is it natural selection? And of course, tanagers are wonderful because they have many species that are sexually what's called dichromatic, meaning males and females are different colors, and sexually monochromatic, meaning males and females are the same colors. Here, for example, is Cherry's tanager, or Rampicillus costaricensis, showing that in many species, when males and females are different colors, usually it's the male that's the more colorful one, and the female that's the less colorful one. But how did they get that way? Well, we might have, in some species, we have males and females that are both kind of drab, and some species, males and females that are very colorful. And so how did they get that way? Well, kind of the prevailing hypothesis for a really long time is that you start off with an ancestor where males and females are both drab, and then sexual selection acts on the male to make him more colorful. And so we could think of this as the change really being driven by sexual selection. However, research in the last few years has recognized that actually, and the alternative is that it's actually might be strong natural selection stronger on the female. If you have a male and female that starts off being colorful, maybe natural selection is the stronger force, making females be less colorful. And so there's been kind of some controversy in understanding, okay, which of these is more common? Actually, my master's advisor, Kevin Burns, was one of the first ones to show that in tanagers, it seems to be perhaps female-driven change evolution of sexual dichromatism. But there are some issues with the when he did it, which was great at the time. Looking only at tanager genera instead of tanager species, there's a lot of variation among species. And then the other thing was how plumage was measured. And it has to do with color and how we measure color and what color is. So, so when Kevin would scored tanager color, which was totally fine to do, he would look at birds and say, okay, this one's colorful and this one's not colorful. However, there's a problem with that. And that has to do with how color is perceived. Well, let's take a step back and talk about what color is. Well, this is the electromagnetic spectrum, which ranges all the way down from very small wavelengths of light in the ultraviolet all the way through what we call visible light, because that's what we as humans can see. And then in longer wavelengths into the infrared, microwaves, radio waves, etc. So as humans, we have what are called trichromatic visual systems, which means in our eyes, we have 3 cones that perceive different wavelengths of light. These are roughly red, green, and blue, which you think about RGB color space, red, green, blue. That's because our eyes have those 3 receptors. And depending on where the light is, these receptors are going to be activated in different ways. And so, for example, if most of the light is over here, we're going to perceive something as being more blue in the 400 wavelength approximately. If something's over here, we're going to perceive it as being more red because this cone is really going to be activated. So which proportion of these cones is activated by a particular object is going to send signals to our brain that are going to interpret that as color. Now, why am I talking about this? Well, birds actually have different visual systems than humans. Birds have what's called a tetrachromatic visual system, which means they have not 3 cones, but actually 4 cones. And that 4th cone actually resides in the ultraviolet part of the spectrum. So we can only see down to 400 nanometers, but birds can actually see down to 300 nanometers. And so there's birds can see a whole range of colors that we simply just can't perceive with our eyes. When we think about how to describe color, this is a human color space. We could think of it as being 2D. And you can place a color depending on how stimulated each of these 3 different cones are. And I would, point to this and say, okay, this one has approximately this hue and this chroma. However, birds, because they have 4 different cones, they have what's essentially like a whole nother dimension of color space. So not only can they, put a point there, but actually, birds have colors like red plus ultraviolet, green plus ultraviolet. There have been behavioral experiments that have shown that this is actually true and that birds can distinguish these things, which is really interesting. And so one question that I'm constantly asked is, can you please make bird goggles that can show me what a bird sees? So I'd love to see the world as a bird sees. And unfortunately, the answer is no, because our brain is simply not programmed to accept that, not just to see UV light. I can show you where UV is in a bird with a UV camera, but we don't have that dimensionality in our brain to be able to process that. So the best I can do is take away one of our channels of light. Here I've taken away red, and you can see this painted bunting looks quite a bit different when it's missing all of its red. So just imagine what it might look like if we had a whole nother channel of color that you could turn on.
So I have a question.
Yeah.
So could that mean that the females could actually be, if they went drabber, then they could actually be brighter? Maybe.
Yeah, so you were onto something. So that what you are suggesting is that perhaps we need to study bird color not from the human visual perspective, but from the bird's visual perspective, because there might be things that we're missing.
They could actually be really bright and colorful.
Exactly, they could be really bright and colorful. So how do we get around this? Well, I have a device called a reflectance spectrophotometer. You can see this little probe, which I can use to measure color. I do it off of museum specimens. You could do it off of live birds as well. Just a lot harder to capture, 382 species of tanagers from throughout Central and South America. And so with this spectrophotometer, you can see this set up here. This spec is actually this little box. This other little box is a light source. And so light goes from the light source through this, probe fiber optic cable, hits the specimen, goes back into the spec, which goes into the computer. And that gives us an objective way to measure what wavelengths of light are being reflected off of these bird specimens. And so for my master's, instead of doing that grand expedition throughout Central and South America, which would have been great, but would have taken me, 10 years or 20 years or however long, instead I did an expedition to different museums to measure all of the tanagers that I could from their collections. In total, I captured over 90,000 of these reflectance spectra from 361 species of tanager. And to your point, there are some patches that actually are essentially ultraviolet colored. So this is a glistening green tanager. It has both shoulder patches and ear patches that look just kind of boring gray to our human eyes. But this is what's called the reflectance spectrum. So the wavelength of light is on the x-axis, the percent reflectance is on the y. So the higher of that, the more reflectance is coming off of it. And 400, again, that's the UV cutoff. So you can see virtually all of the reflectance is in the UV part of the spectrum. Now, totally ultraviolet colored birds are actually pretty rare. Usually the UV is associated with either also reflectance in kind of the blue violet range, or sometimes you'll have UV plus yellow and red in some of the kind of red or orangey birds, but they do still exist. So it just highlights the importance of studying things from the bird's perspective. So the first question that I wanted to ask with this was, does this matter for studying sexual dichromatism? Are there things that we can't simply detect? And so for all species of tanagers with field guides, we scored, do field guides describe these as being the same or different? Males or females being monochromatic or dichromatic? And when you score them that way, it turns out they're about 50-50, about 50% dichromatic, 50% monochromatic. However, with our data set, when you apply the avian visual, a model of avian vision to these objective measurements, it turns out that it's actually more like 94% dichromatic, only 6% monochromatic. So there is a lot of variation that we are missing with our visual system, which means that whenever we measure color, we really need to be analyzing them according to the birds. Yeah.
What about bird predators that might be part of the natural selection?
Ah, great question. Yeah. So, you know, some people have really gone on, like, once we discovered that ultraviolet color was in the plumage, people were like, is that a secret channel for birds to communicate with each other? And actually, the ability to see UV is actually the ancestral state, meaning that a lot of lizards can see UV, fishes can see UV. It's actually mammals that lost it when we were nocturnal, our ancestors long, long ago. Most mammals actually only have two types of cones. You might have heard that your cat or your dog can only have black and white vision. That's because they only have two cones in their eyes. Humans, you might have noticed that one of our cones was not very different from the other one. Back in, I think it was in the primate lineage, the gene for one of those cones duplicated and got a little bit different. So that restoring our trichromatic vision from just having two cones. So we still can't see quite as well as things that ancestrally had those three cones that are more evenly spaced on the kind of electromagnetic spectrum. Yeah.
I'm going to add here, I had read this a few years ago, that some birds, the male and the female, look exactly alike, and people have asked, how do they tell each other apart?
Mm-hmm.
And this was one of the theories, that in the UV light, they probably look different.
Yep.
And there's colors that we can't see. So that would be another thing. Yeah.
Exactly. The UV color. You know, I didn't talk about this, but birds also have these little oil droplet filters on their cones, which allow them to distinguish between similar colors better. So there's like even more to it. Okay, so now we know plumage must be quantified from the avian visual perspective. So how do we go from just getting these distances between things to actually quantifying being colorful, essentially? Well, let's take our friend, the paradise tanager, which in many ways has been kind of a mascot of a lot of the research that I've done. This is what the reflectance spectra from its major patches look like. And then we can plot those in the avian tetrahedral color space. And once you have all of the points in there at the same time, we can do things like measure the average distance between them, which tells us how contrasting the points are. We can see how much of this space is occupied by all the points. We call that color volume. And so with these measures, we can actually quantify what's colorful or not. Alternatively, just to show you that these measures actually do matter, so this has a very large color volume. This has a very small color volume, the small ground finch, Darwin's finches, actually tanagers, not finches. Another fun fact. You can see its reflectance spectra are all flat, which is very much how black spectra usually are. And they all pretty much are in the same place. So this is a bird with a very small color volume and very small measurements. Okay, so back to tanagers. Tanagers are really great for studying the evolution of color. And here's what all of their patches that I measure look like in that avian tetrahedral color space. Just beautiful. Okay, so back to studying males and females and using them to kind of disentangle natural and sexual selection. So the first thing we wanted to do is ask, do males and females tend to be equally colorful, or are there tend to be differences between them? If they were equally colorful, we would expect all of our data to fall out on this one-to-one line, where if you have colorful males down here, you also have colorful females and vice versa. However, another possibility is maybe males are usually colorful and females are less colorful, in which case we would expect all the points to kind of be down here, or vice versa. Maybe females are the colorful ones and we would expect all the points to be up here. So what do our data actually look like?
Yeah.
Well, you can see actually for the most part, males and females fall out pretty nicely on this kind of one-to-one measurement where for the most part, males and females are equally colorful. But when they do fall off of this line, it tends to be with the males being more colorful than the females. So, you know, this is not surprising because that's generally how we think of bird colors as operating, but it is nice to see our expectations being met by these, you know, kind of fancy quantitative measurements. Now, what about now back to this ultimate question was, is it sexual selection driving males to be more colorful or natural selection driving females to be less colorful? Well, the next thing we did was actually calculate sexual dichromatism using measurements between males and females and seeing if the male plumage is correlated with it or the female plumage is correlated with it. If males are correlated, then that suggests that it's probably more often sexual selection driving the evolution of sexual dichromatism. If female plumage is correlated, it's probably female plumage. So what do we find? Well, we do find a male correlation. You can see there's a lot of noise here. There's a lot of points all scattered about, but there is a significant correlation, and there's not a female correlation. So contrary to what my advisor found as part of his dissertation, when you actually quantify plumage from the avian visual perspective using species measurements. Perfect. All right.
Yay.
So now we're moving from studying color by itself to understanding the components that make up color. And so what do we mean by that? Well, feathers are arranged where you might have different colors on different parts of the body, which can be important. Just an example here, these are some hummingbirds that you see, and you might notice if you look at a lot of hummingbirds that they tend to be colorful on their crowns and their gorgets, and then the rest of them tends to be less colorful. Not to say that they're not colorful and they're not exceptions to this rule, but typically the crowns and the gorgets are the most important patches for signaling. And so that suggests there might be kind of different pressures on different parts of the body. Just for fun, these are some sunbirds, which are the ecological equivalent of hummingbirds in Africa and Asia and Australia, because hummingbirds only live in North, Central, and South America, but they've evolved very similar patterns for doing similar things, which is very cool. So then we can get even smaller and think about feather macrostructure. What do I mean by that? Well, here we could think about the components of a single feather itself. Here's a feather. We could think of, the fact that the feather is divided up into different parts. So there's what we call the pennaceous part, which is what is visible when you're looking at a bird. Then there's the plumaceous part, aka the downy part of the feather, which is more commonly associated with keeping the bird warm. Off of these, we have feather barbs. So we call that feather macrostructure. You can go zoom in even more, think about feather microstructure. And so that's, if we look at the individual feather barbs, you'll notice that feather barbules come off of those. And so that there can be variation amongst the feather barbs and the feather barbules as well. Just the feather barbs, the center is called the ramus. And finally, we can zoom into that and look at the inside of feather barbs and feather barbules and look at how the molecules are arranged, which is important for producing those colors. What we call structural colors actually are dependent on the arrangement. Here's like a cross section of the feather barb. The inside of the barb, we've got kind of this first a melanin layer, which is going to absorb a bunch of light. Then there's this layer of molecules that are arranged in what's called a spongy pattern, which is going to refract particular wavelengths of light. In the case of this bunting here, it's refracting blue light. And so that's how colors like blue and purple are created. Iridescent birds like hummingbirds, so that's where the angle changes, or the hue changes dependent on the angle, have these kind of layers that also refract light in a particular way. So that's dependent on the arrangement of the molecules inside of the feather. This just shows iridescent birds, structurally colored birds. And interestingly, non-iridescent structural color is usually found in the barbs. You can see this is a blue jay feather, and so this is the central rachis in the middle, and you've got these, you can see the bright blues and the barbs, whereas the iridescence is actually in the barbules. So if I had a picture of a peacock feather, which I should put into this slide, you would see that it's not the barbs that are colorful, it's the barbules. And finally, we can get just one final layer, which is actually the molecular composition of those molecules. And so in particular, what people pay a lot of attention to are the pigments. And so pigments are what produce most of our browns, our blacks, our yellows, our reds. For example, in this cardinal, so pigments absorb light. So structure refracts light, pigments absorb light. So depending on which pigment you have and what structure you have, you can produce these colors. There are two main pigments. One is called melanin, which produces blacks and kind of browns and rufous colors. So shown by this nice American robin or spotted toady or this crow. The other most common pigments are called carotenoids, which produce yellows, oranges, and reds. Think carrots, for example, produce oranges. And so it's really these that are producing a lot of what we see. Oh, and I would be remiss if I don't mention green, which is mostly produced by carotenoid pigments plus blue structure. So there's actually only one bird that has a true green bird, which is my favorite bird, which is the turaco. If you ever seen a turaco, beautiful green birds down in Africa, but actually have these very unusual green pigments. Okay, so a lot of research has been done kind of on different layers, but what I'm really interested in these days is in feather microstructure. And how did I get interested in feather microstructure? Well, when I was a PhD student, a fellow PhD student named Cody McCoy, who was talking about her undergrad research she had actually done, describing what's called super black feathers. And so what this means is these are scanning electron microscope images of a regular black feather and a super black feather. So in a regular black feather, you have a barb and the barbules are coming off of that and they essentially lay flat. They're just all flat to the plane. Whereas in super black feathers, this is a bird of paradise, which of course has the most extreme microstructure of all feathers as they have the most extreme color patterns. The barbules are angled and they actually, in this species, actually have little projections coming out of them. And what really gets you, and so this is basically these super black feathers are absorbing more light than the pigment can alone. So one of the steps in using a scanning electron microscope is doing what's called sputter coating, where you actually are coating your material with a very thin layer of gold. When you coat regular black feathers with gold, they look gold. When you coat super black feathers with gold, they still look black. If you've ever heard of Vanta black paint, it's very similar to that type of paint. It's just actually birds did it first. So why was this, when Cody gave this talk, I mean, of course it's really cool. I thought it was really cool, but what brought me back to this bird that I had measured as part of my tanager data set, this is Ramphichelys carbo, or the silverbeak tanager. And why her talk reminded me of this bird was that I can look at this bird and clearly see that it's red. It's a deep maroon, a gorgeous bird. However, when my reflectance spectrometer essentially reads all of the light off of this bird as black, which is a tiny, tiny peak in the red. So actually most of the Ramphichelys readings were dropped from the data set because they didn't pass my threshold of having a 5% reflectance, which is necessary for doing analyses. So that had always been a puzzle to me. Why is this bird reading so dark when I can clearly see that it's red? So after Cody gave her talk, I said, hey, Cody, I have this bird. It's really interesting. Do you think maybe it has that super black structure plus carotenoids inside? And Cody said, maybe, let's find out. So we did. So this actually started what ended up being a several-year collaboration between the two of us and some other folks at Harvard where I was, and we ended up studying kind of both male and female Ramphichelys tanagers. There are 10 species in this genus. And in this group, they're mostly males have this either really nice black, super black color with beautiful reds or yellows associated with it, and females have generally kind of similar patterns but are usually a little bit less vivid than males. And so what's one thing that I will mention is back to carotenoids, which are producing those yellows, oranges, and reds. People like to study carotenoids because they're thought to be honest indicators of a bird's quality. you look at house finches, we could look at we could have looked at house finches in the garden when it was still light outside. And you might have noticed that some are really, really red, some are more orange, some are maybe even yellow. So there's really a variation in house finch color. And house finch is actually kind of like the bird people research to study this. And they've shown that the redder you are, kind of the better a potential mate you are. So the more that females are going to be attracted to. And that's because you have more carotenoids in your plumage. And carotenoids, they're not only important for making your feathers red, but they're also important in your immune system. And so if you can put them in your feathers, that means you're healthy enough to not reserve them for yourself, for your immune system. They're antioxidants. They also have to be consumed. Birds can make their own melanin, but they have to eat carotenoids from their diet. So also shows you're a good forager. So it's not the redder you are, the higher quality you're going to be as a mate.
Allison,
Yeah.
I think habitat has a lot to do with it because the house finch in urban areas aren't nearly as red or orange as those in the native areas.
That is a great point. And that's actually a future project that I have in my head as a potential community science project to crowdsource that, because nobody's ever documented that before. So
I've observed
I
it.
agree. Yeah, and actually there's a lot of house finches are native to the West Coast. They're introduced to the East Coast. Interestingly, there are much more red house finches on the East Coast, possibly due to the fact that when they're introduced, probably the most beautiful males were the ones that were shipped as part of the pet trade. So there might have been some genetic quality control. And then they're also introduced in Hawaii, where there are actually many more yellow and orange ones there, which could again be due to kind of habitat quality. So they're very interesting birds for many reasons. And so back to Ramphichelys, well, when we're studying males and females, we expect, even though we expect the males to have more carotenoids, to have more of these expensive pigments in their feathers, because they're so vibrant and colorful. And so what we wanted to understand was it variation in the carotenoids, which we actually used a technique called liquid chromatography mass spectroscopy. It's big words, LCMS, but just know it allowed us to understand what type of carotenoids there were. And then we used scanning electron microscopy to also look at the microstructure of both males and females in this group. So what did we find? Well, this is what the carotenoid results look like. I know this is a lot, but just know that the darker the box, the more of that type of carotenoid, and then they're grouped into essentially carotenoid families with different isomers if you're a chemistry person. But the point here is that if you squint at both these male and female results, they look largely the same, right? Very much similar. And we can actually look at this in correlation, and they are statistically similar. So it's not that the carotenoids are different between the males and the females, which was very surprising to us. We did not expect to find that. However, so then we turn to the microstructure. The first thing we learned was that females generally have microstructure that looks like a normal feather. So this is a feather barb. The barbules all kind of lie flat. The black part of these males have this super black feather microstructure. So the barbules are angled at about 40 degrees, capturing all that light. And one thing we found that we did not expect to find was that actually the red feathers had a lot of variation in microstructure too. So we found that the really red feathers had these expanded, really, really fat feather barbs. This is a cross section of a feather with tiny little angled barbules. Our friend Ramphichelys carbo, the one that started all of this, in fact, did have super black feather structure plus carotenoids in the middle. So that was really exciting to find. And then we also, some of these other red birds had these kind of fat, but also oblong barbs with angled barbules. And interestingly, the females all kind of fell out together, which means that they didn't have a lot of variation, but the males had a ton of variation. So it wasn't carotenoid, it wasn't pigments where they varied, it was actually the microstructure holding pigments. We did some very fancy kind of simulations. All this is showing is that if you simulate feather barbules becoming more angled, reflectance drops. So this is basically saying, yes, that makes these feathers be extra dark. And then what this is saying is that, okay, and then if we simulate kind of a large extra barb that has more pigments in it, like the male feathers did, there's more optical power, AKA more color coming off of that. And so really, you know, these differences were meaningful in what these feathers will look like, which is very cool. So we found that microstructures can have a major impact on the optical properties of these plumage patterns, and they do vary between males and females, but carotenoids did not vary. So we kind of pitched this in our paper as maybe males are dishonestly signaling. So they're making themselves look brighter without putting the expensive carotenoids in. But we'll see with future research whether that holds. You know, one question I have is, okay, if you change your microstructure, what else does that do to your feather? Feathers do many things, as you'll learn in one moment. So, as I said, you know, I really used to be focused on color and pattern, and now I'm getting more interested in kind of the components of how those colors and patterns are produced. And if we come back to this question, are birds' plumage patterns shaped by both natural and sexual selection? You know, this is really actually a simplistic view, because it's not just about what color you are in terms of being cryptic or being colorful, because as I hinted at, feathers do many things. Another observation that you might have, if you have birded in a lot of habitats and you've seen song sparrows, maybe all over their range, you might notice that some song sparrows are really pale in some areas and really dark in other areas. The same with fox sparrows. That's true of many birds. That's actually what we call an eco-geographical rule called Gloger's rule, which says that birds that live in hotter, more humid environments tend to have darker feathers, and birds that live in less humid environments tend to have lighter feathers. And so that says that the environment also might be shaping feathers. So my friend Ryan Terrell and I, during COVID, we actually wrote a whole review on all of the functions of feathers. We identified 26 different functions, 26 different things that feathers do, ranging from, of course, flight, allowing birds to fly, allowing them to maneuver, to swim, to brace themselves, to allow them to keep them warm. Some of these are perhaps not surprising. Others might be. If ever, you know, protecting them from parasites, lining nests, helping them to forage, reflecting sound potentially, transporting water, and then a lot to do with communication. So mate choice, social selection, maybe mimicking other species, maybe distracting some species. So feathers are doing all sorts of things. And one other thing that we started to do was, okay, if we know feathers are doing these things, as I mentioned, you know, feathers can vary on the body. So we might expect certain body regions to be associated with certain functions. And people have actually started to look at what part of the feather is responsible for some of this variation. What do I mean by that? We just think about color and pattern. And I know these words are very small, so don't worry about reading the words, just think about the colors. There's a lot of communication, especially in the head and the ventral region, kind of the front of the bird, that is typically used in social communication, whereas the back is maybe more typically used in crypsis, because that's what a predator is going to see from above. Whereas the wings and the tail are really important for flight and for foraging. And so that's overall color pattern. But we can do it, you know, people have associated microstructures with thermoregulation, because actually how thick your barb is might allow more light to pass through, more heat to pass through and get to your body. Think about downy proportion of feathers. If you go up in elevation in the Himalayas, birds have more down to their feathers because they need to keep warmer. Just a few other examples. I really, Ralph Schreiber in this paper in 2006, they found a frigate bird that had washed up on the coast of, I think it was either New Hampshire or Maine, somewhere on the eastern seaboard, but it was an albino, and the feathers were just destroyed because melanin is actually really important for strengthening feathers. And so if you're a seabird that's exposed to salt and you're flying a lot, your feathers aren't strong enough to withhold that. They're just going to be destroyed and you're not going to be able to fly anymore. So melanin is really important for feather durability. Another example, I just saw these this weekend. I was camping at Silverwood Lake, but we have phainopeplas. And so you might think, if you're a bird that's in a really hot area, perched out in the sunlight, wouldn't you want to be light colored? Because dark colors absorb more heat. However, it's been shown that that Phainopeplas actually use their dark plumage to capture the heat and not allow it to get to their body. And so wind can disperse it. So it's almost like wearing a robe in the desert, as some desert peoples do. Finally, one of my favorite examples is the sand grouse. Anybody here heard of sand grouse? Yeah, they're super cool because the males actually will use their belly feathers to transport water. So they nest, dozens of miles out in the desert. Like we're talking the Sahara Desert, really, really hot places. And the males will actually go to the watering hole and soak up water with these very specialized belly feathers that are evolved this interesting helical structure to capture more water and ferry them out to their young. I'm excited. I'm just actually starting a collaboration with an engineer to study these feathers because they actually, have some potential engineering applications as well. But if your belly feathers are good at sucking up water, what are some of the other things maybe that they're then not good at? So we really need to understand, when we're studying these things, understand the trade-offs of these different functions as they're evolving. So I just wanted to end with some of the other people that we have in the ornithology department. Many of you mentioned Kimball Garrett, who did retire last spring. And so this is Young Hassa, who's his replacement. Not that one can replace Kimball, but Young is amazing and is filling the function of collections manager.
Which one's you?
This is me, and this is me. And
I
so
was thinking maybe the one in the second one to the right.
This one?
Is that one you?
This is me.
The
Yes.
one's the second one to the right.
This one?
Right.
This is not. This
In other words,
is my friend.
second to the left.
This
I mean,
one.
yeah,
Yep.
that was you.
Yep. So that's me too.
And that's
So-
you too.
And this is yep. So I was at the bird meetings this summer, and I had my whole lab together for the first time, which was really exciting. And so I just want to highlight some of the interesting research that they're doing. This is Jessie Salter, who's a postdoc, who actually is going to be starting at Santa Monica College in the fall, teaching there. But she studies bobwhite quails and how their feather microstructure is co-evolved with the feather lice that's eating it across their range, which is really interesting. So she's actually combing museum specimens that are up to 100 years old and getting feather lice that were attached at the time they died. So you never know what you're going to find on these museum specimens. This is Maeve Secor, who was actually in the LA Times just yesterday she was banding at Bear Divide. I don't know if any of you saw that article. She's really interested in downy feather structure and how that evolves, not just in the Sierra Nevada, not in the Himalayas, but in the Sierra Nevadas. And as the climate changes, do we see any evolution of the downy part of the feather as birds either change their range or do not? This is Rosalind Price-Waldman, who's studying Tangara tanagers with me. And we're studying how did this group, which is arguably one of the most colorful groups of birds that has evolved color incredibly quickly, how do they produce these colors and how has it evolved through time? And this is Jackie Childers, who actually just started 2 weeks ago, who's another postdoc who's studying weavers and is looking at feather color, egg color, and nest shape and how that all relates to the environment that these birds live in. There's a lot of exciting things happening in the department. A lot of really cool science being done, but hopefully through this talk, you've been convinced that to understand the evolution of plumage patterns, we need to understand the mechanisms that produce them and also understand them from the bird's perspective. And finally, the evolution of feather coloration can only really truly be understood in the context of the many different functions that feathers perform. And so I just want to end with some acknowledgements for my many collaborators for this research that I presented. Of course, the museum collections that have maintained these amazing resources to study, and finally my funding sources for funding various research that I presented. And I'm happy to take any other questions you all might have.
Oh, I think I probably might have a question for you.
Yeah.
No, somebody once noticed when I drink so much Red Bull, they said to me, "Are you trying to turn yourself into a bird?" And I was like, "Why do you tell yourself because you're drinking so much Red Bull? Because you know what? Red Bull gives you wings, doesn't it? Does it really turn you into a bird?"
No, you'll have to keep drinking it.
Somebody, because I saw my coworker, somebody said, "It's like, are you trying to turn yourself into a bird?" When I kept drinking Red Bull.
I thought you were going like, are you trying to be a bird because of high metabolism?
Yeah.
Maybe that's what my coworkers meant.
Yeah,
That might have been what my coworkers meant, though, when they said that.
yeah,
Because it's a commercial, what it says is like Red Bull gives you wings.
that's right.
Remember?
Yeah,
in
any
a
other
commercial.
questions anybody has?
Okay.
What's the most interesting thing you do with your job every day?
Oh.
Would you say? Or not every day, but just.
Well, the thing I love about my job is that I get to do different things every day. So, you know, some of it is working on this research. Today I was just working on, you know, doing some computer programming stuff, but I love that other days it's to, you know, talk to groups or do outreach events or do, you know, give a talk at university. So the thing I love about my job is that I get to do different things all the time and work with so many wonderful young people as well as older people too.
Oh, that's good.
Yeah.
Since it's not a tanager, is there a name change in the works for the Western tanager?
No, you know, they tried to get it changed to the the Ferenga, the Western Ferenga, but that was a no-go.
Yeah.
So, you know, people have non-tanager. People have argued that tanager then is more like kind of about the ecology, like finch. There are many things that are called a finch that are not in the finch family, or sparrow even. You know, there are many families of sparrows. So tanager isn't necessarily a taxonomic classification.
What makes a tanager?
Well, you know, that's the other thing is they just put all the things in the tanager family before that they didn't know what to put anywhere else, right? So, yeah, I don't know if you can really argue that, but they do.
So if there's one place you can go to see birds, where would that be?
Well, I just did some field work in Colombia last September, capturing tanagers that were actively molting their feathers, which was very exciting. I didn't have time to tell you all about that. I'd say elsewhere in Colombia would be a high on my list. But I would also love to go to Africa because, as I mentioned, turacos are my favorites because they have the green pigments. And there's just that I have never been to sub-Saharan Africa, so there's like a lot of birds. Turaco, turaco.
Go to Uganda.
Uganda, yeah, I'd love to go to Uganda. So I don't know. There's so many places in the world with wonderful birds. Oh,
And
yeah, we have a picture.
Allison,
Yeah.
hey, you know one other thing. In one of my previous jobs, I used to have a coworker who used to like to use paper to make a bunch of birds.
Uh-huh.
I had a coworker that does that all the time.
That's
And that time he was actually the one that was training, my startup trainer, and he was just doing that all the time. And he would ask me, "Am I boring you out by doing all this?"
yeah, you know, actually a thing that I love about birds is just
He
how
was making them with paper.
you can use them in art. So whether it be making them out of paper, I actually am a fiber artist as well. I do embroidery and weaving and are very much inspired by birds. So I'm hoping to actually do more either at the museum or outside of the museum, kind of thinking about birds as art, birds in art, combining like the biology of color with color theory.
Because that time he was actually training me on how to process the election applications, but at the same time he was spending a lot of time making birds out of papers. And then he goes like, and then he's just making a bunch like sitting over. Am I boring you out by doing all this? Even.
Yeah.
There's a bunch of wild parrots.
Yeah.
Run around that look green to me.
Yeah. So all those birds, so parrots actually have a special parrot pigment, which are called psittacifulvins, that make them red and yellow. But it's also, so they have blue feather structure plus these yellow pigments, which makes them look green. But if you were to grind up a parrot feather, the dust would be yellow because you're destroying the structure, but the pigment's still intact. Think about finger paints.
Yeah.
Looks like we have fabulous.
Any questions from Zoom? Yeah, we have a question. I don't know, Gayla, if you want to ask. I can also.
This is a little, hi, this is a little bit off. Actually, I saw your presentation at another bird club and I just thought it was fascinating and I still didn't catch everything you said.
I know, it's a lot of
There's a lot.
information.
It was great, absolutely wonderful. One of my favorite presentations I've seen. But do humans emit any kind of ultraviolet rays that a bird can see that humans cannot see?
You know, that's an interesting question. Our clothing certainly does. And so that's one reason why, oh, sorry. The question is, do humans emit ultraviolet rays that birds can see, but humans can't? Yeah, so clothing can. And so especially like white clothes are incredibly, usually there's a lot of UV, depending on what kind of pigments are in your clothes. Humans, I don't believe that like our skin and our hair is particularly UV emitting, but it's mostly clothes for sure can be very bright in UV. You know, one fun UV fact of, you know, how birds use it is actually kestrels. They can see falcons can see a lot of UV, and actually like the urine of some rodents is very reflective in UV. So they'll just follow the urine trails, which
What color clothes do you wear
when I go birding, you know, I generally like browns and, you know, greens, and I won't wear my bright pink clothes.
Someone also commented that maybe the
Makeup,
like colors,
nail
no
polish.
Can I add one more thing? Is
Wow, yeah,
My
I love
pet-
it. Yeah.
my parrot can tell the character of somebody, and they're, you know, they're 20 yards away. And I'm wondering, how can she tell? Are they, you know, like, are people having auras? I mean, is there a color that they can see that we can't see?
So the question is, how can parrots determine the character of a person from really far away? Can they see people's auras or their energy? And to the best of my knowledge, no. But I would say, you know, there are probably behavioral tells that people do that animals can pick up really well, especially very visual animals like parrots, for example. The parrots are incredibly intelligent. So my guess is they can pick up on body language and some of these other things that maybe aren't as obvious to human. Yeah, to the best of my knowledge, I don't know if people do have auras. Maybe they do. Maybe you never know. But yeah.
Thank you.
I have a related question.
Yeah.
Why would birds like parrots be so good at detecting human body language?
Yeah, so the question is, why are birds like parrots so good at detecting human body language? And that could be, you know, well, I would say perhaps, you know, they're really good at detecting bird body language as well. And maybe it's actually easier in humans because of how expressive we are. You know, if they're birds that have been raised by humans, just like cats or dogs are actually really good at detecting human body language as well. You know, that's their main form of communications. They're picking up on small cues that maybe we won't, because we're used to communicating by words. Although, you know, I think people actually pick up on more body language than you necessarily think about too. Yeah, but
Well,
my guess is it's just behavioral similarities and or exposure to people.
I
A lot will
asked,
be chemical. I would say that a lot of it is pheromones as well.
Yeah, so that one comment is a lot of it may be pheromones. And, you know, for a long time, people thought that birds didn't really smell or didn't really smell much. I think there's much more recognition that a lot of birds actually do smell quite well, and not that they smell, but they have the ability to smell very well. And so that's actually a very active field of research right now is bird olfaction capabilities.
I asked Dr. Irene Pepperberg, who does the research on African greys at Harvard University, about how my bird can tell if a person owns a bird or not, or they're an animal lover. And she told me they're just very, very sensitive.
Yeah, so the comment was, she asked Dr. Irene Pepperberg, who's actually a professor at Harvard University, about the fact that her parrot can tell really well how a person is, and she said that birds are very sensitive. So I think that is true. Parrots are very sensitive to people.
Has research, so one of my colleagues did research on birds and like preventing bird collisions
Yeah.
to airplanes. And they had it was a calcium luggage. They had it with lights
Mhm.
and trying to see like how far they could see, like in the periphery. Has there been like research done that was like a while back
Mhm.
for like adding like UV or other colors on planes to prevent those types of collisions? And I don't know
Yeah,
if you've
I
read
haven't
up on that.
I haven't read much about like plane collision preventative
But even
type
like
research.
For
But yeah, windows,
windows.
people are, I know there's a company who is doing active research on kind of ultraviolet decals to see how well those might work with birds. So I don't think there's an answer yet. Nobody's shown that ultraviolet decals work well enough to make them like the solution, but people are studying it for sure.
And last question, are there
Yeah.
any colors that like the birds don't like that you've seen that you're like they're not used as much in plumage, or I don't know?
That's so. What colors do birds not like or not use as much?
Prefer- Yeah.
Well, there are colors that are harder for birds to produce, like especially very structural colors. You know, those have evolved in some families, but not other families. So it kind of depends on, do birds have access to the capability to produce those colors? And then I would say it really depends on like habitat matters a lot. Where you are matters a lot. So there's not necessarily a single answer. It's really very species specific. Yeah. There's certainly colors that are like rare in birds. Like, yeah.
Thank you.
Yeah.
Well, a colleague at UCLA
Mhm.
in administration, she was a birder, and they have these really tall doors that are glass.
Yeah.
And she got tired of seeing dead birds, and so she got a custodian to put up a predator outline,
Mhm.
and that solved the problem.
Great.
Wow.
Yeah. Yeah, sometimes just like putting something on them so that it breaks up the reflection. The museum, if you've ever been there, has this kind of big glass building that we finally got them to put bird stickers on the bottom to help prevent bird strikes. Yeah, it's a big problem. Like, those glass fences, I hate those that people.
Are you talking about at the actual museum?
Yeah, at the museum. But like, yeah, houses overlooking the ocean and stuff. I grew up in Dana Point, so not too far down the road, and a lot of glass fences. Glass fences, not so good for birds.
At least during migration season. you
Yeah,
know.
well, and other times too.
Yeah, that's true.
Yeah.
Yeah. You had talked earlier about the house finches, how that some of them are red and some of them are yellow and orange, different colors. And I'm wondering, one of the things you said was it might be what they eat.
Yeah.
So what is it that they eat that causes them to be red?
Yeah, so so a lot of fruits, but also vegetal material and insects have the carotenoids necessary. But most of the what are called dietary carotenoids, which birds can just eat and prick directly in their feathers, those are yellow. They actually have to metabolize them to make them red. So yeah, it's they have to like enzymatically turn them from the yellow carotenoids to the red carotenoids, and that can actually be a very complicated process.
Okay, so it's not like the flamingos that eat the Gulf shrimp and all that stuff. Okay.
Yeah, but if you were to raise a house like any carotenoid color bird in captivity without access to any carotenoids, they would not produce that color.
Okay.
Yeah.
Because I see everything at my feeder, I see, it is mostly house finches, and I see all sorts of color variations, and some are a lot more red than others, and some are yellow morph, and
Yeah.
very interesting.
Yeah, so some of it has to do with diet, but as I said, because they are metabolized, that's also expensive. So some of it is genetics, some of it is how healthy they are. There's a lot of factors that go into like, are they red or are
I
they not?
always thought it was just that the less red ones were eating too much junk food.
Yeah.
Junk food, we eat much junk.
Right. Any last questions? I think.
Oh, somebody's got to-
Thank you all for joining
Sally,
us.
Yeah.
Sally up there, she's got her
I
hand up.
had a question. Sometimes when I go out looking for birds and I'm wearing a particular color shirt, I think birds of that color are attracted and come out to look. Is that just silly of me or just maybe something
Yeah. So the
happens?
question is when she wears a certain color of shirt and birds of that color might be attracted to it. And that is actually a known phenomenon. That's one reason why people think that birds that are more commonly fruit-eating birds or nectar-feeding birds tend to have bright colors because they're inherently attracted to brighter colors. So I wouldn't say it's probably a 1-to-1 ratio of birds of that colors are attracted to that color, but there is something to that because there is probably a reason that, for example, male birds are a certain color, females are attracted to that color. And so, yeah, especially during breeding season, if the males have like a ton of testosterone and they're really like hyped up, they might be attracted to all the bright colors, right? So.
Oh, good point. I didn't know that.
All right.
I think that's it. Thank you all for
Yeah.
coming.
Yeah.